Hello, everyone. Very good evening to all of you. I hope I'm audible. Please quickly confirm in the comment section. May I have a look at our audience today? Welcome to all of you. Very warm welcome to today's special mentoring session on NGS careers, where you can get all your questions answered. Okay, great to have you all here. Welcome Anjali, Gunjan, Jyoti, and all others. And I'm really happy to see so many students, researchers, life science professionals who are joining us here today. Now, honestly, if you are from biotechnology, microbiology, genetics, bioinformatics, pharmacy, zoology, botany, biomedical science, or even if you are simply confused about what skill to learn next, then this is for you, because today we are going to talk about one of the fastest growing domains in life science, and that is NGS and multi-omics data analysis, right? And more importantly, what careers actually exist in the field, which skills are companies and research labs expecting, whether coding is compulsory or not, can non-bioinformatics students enter this domain, what salaries and opportunities are available, how do you start practically, what tools you should learn first, how do project and internships help in getting these opportunities. So these kind of questions students keep asking us every day. So we thought, why not talk in a separate session about all your pain points. So today, instead of generic internet answers, you will hear directly from the experts who are actively working on real world projects in this domain. So make sure you stay till the end, because many important career pain points will be discussed. You will learn not only from your own questions, but also from others' questions, which may be equally helpful to you. And at the end, we have a surprise gift resource for all our attendees who have joined us today that can genuinely help you start your journey in this sector. So please stay connected throughout the session. Now, quickly, let me give you the structure of today's session. We'll begin with quick introductions of our expert panelist members. Then we'll open the floor for your questions. We will pick questions from the comment section and direct them to our panel members. You will get your responses, and by the end, we end the session by sharing the surprise gift link with you. So this is going to be highly interactive and practical. Let's invite our panel members to the days for today. First of all, we have Dr. Bhupender Singh, who is currently working as a CRO scientist and AI/ML trainer at Biotecnika. He completed his PhD in bioinformatics from Lovely Professional University with specialization in machine learning and in silico drug discovery. He has close to a decade of experience in bioinformatics and has also worked with CSIR IM Tech Chandigarh, contributing to important scientific projects. His expertise includes AI/ML in life science, NGS and multi-omics data analysis, Python and R for biological data science, computational biology, drug discovery, and chemoinformatics. Dr. Bhupender has authored 20 plus research publications, received around 700 plus citations, and actively mentored students in building industry ready skills in computational biology and omics data analysis. Welcome, sir. We are really happy to have you here with us today.
Thank you very much.
Next, we have Dr. Elamathi Natarajan, who is a bioinformatics neuroscientist at Biotecnika and an expert in bioinformatics, AI, and computational biology. Ma'am brings 13 plus years of experience in NGS data analysis, transcriptomics, multi-omics, and AI-driven drug discovery. She has worked with reputed organizations including ICMR, NIMR, NIPGR, and DRDO, and has contributed to genomics and infectious disease research projects. Her expertise includes RNA-Seq and NGS analysis, multi-omics data interpretation, AI and machine learning in life sciences, Python and R-based bioinformatics pipelines, biomarker discovery, and precision medicine. She is also deeply involved in mentoring students and helping them become industry ready in bioinformatics and AI applications. Welcome, ma'am. Wonderful to have you with us here today.
Yeah. Good evening, ma'am. Thanks for your introduction. Good evening, everyone. Lovely to see you, all of you. Yeah.
Absolutely. It's a gorgeous evening. And our third panelist is Dr. Nilofer K, currently working as a CRO scientist at Biotecnika, specializing in AI, ML, and bioinformatics. She completed her PhD in bioinformatics from MIT Edith University, Pune, with strong research focus on canceromics and AI-driven computational biology. She's previously worked with BDG Life Sciences and CSIR National Chemical Laboratory, Pune, including projects to AI-based cancer research and computational drug discovery. Her expertise includes canceromics and multi-omics integration, RNA-seq, scRNA-seq and proteogenomics, AI machine learning, deep learning, precision medicine, and biomarker discovery, Python, R, Scampi, and Bioconductor. She is published in reputed international journals, presented at major scientific conferences, including NVIDIA, GTC, Silicon Valley, and also holds an Indian patent. We are extremely glad to have you here with us today, Nilofer ma'am.
Thank you, ma'am, and good evening to all the participants, including ma'am. So, welcome all of you.
Yes, welcome once again to all of you. So, we'll quickly have a look about what this domain is. So, we have Dr. Bhupender sir giving us a very quick overview of this area and what is happening here. It's a very interesting session. After this session, we'll be starting with our Q&A. Over to you, Bhupender sir.
Thank you, ma'am. I hope I'm audible. And once again, I welcome one and all of you to this most exciting session on how to build your career around NGS and multi-omics data analysis. So I'll be guiding, rather than guiding, I'll be discussing upon few points which I felt like are important before we actually proceed into taking your questions. So I would like to start with giving some introduction regarding various sequencing technologies and how the evolution has resulted into this particular instance wherein we have this opportunity to maybe produce high throughput data now. Then what are their importances in what all fields this NGS and multi-omics data is actually creating a revolution. Then most important part, how this NGS and multi-omics data analysis is actually being more required and demanded in research labs as well as industries. And then after this, various pointers that why one should actually jump into this particular field. So without any further delay, I would like to start with various sequencing technologies. So as you all might be aware that we had first generation of sequencing, which is referred to as your Sanger sequencing, right? So Sanger was the first person who has actually introduced this sequencing thing into our scientific knowledge, and then the further revolutions came into the picture. But specifically, the Sanger sequencing was having this capability that it was very accurate. So accurate in the sense the base call which is being identified on your query genome or query DNA that may be at particular position, whether it is A or T or G or C. So it is highly accurate comparatively when we talk about other generation of technologies, but it was having some limitations. So those limitations are primarily in the field of in the area of scalability. So these methods were actually not scalable. Then what happened next? Then we had introduction of second generation of sequencing. So in second generation of sequencing, we were now able to perform this high throughput sequencing, or what is referred to as your massively parallel sequencing. As you can see here, which basically this particular second generation sequencing or next generation sequencing corresponds to generation of clusters, and in order to get higher and higher signals, and not only get the higher signals, but here simultaneously parallelly you can actually sequence multiple reads, and that is the reason that the previous generations limitations were overcome, right? Though this was till around 2010, and then from there onwards we saw a miracle wherein third generation sequencing came into the picture, and now we had more sophisticated systems. So these now, as you can see here, were not requiring any amplification process. So as you can see here, in the second generation sequencing, prior to sequencing of your query reads, the amplification is required. But here, in the third generation sequencing, you can directly, pass your reads into your nanopores, and in the real time, you will get the electrical signals, and based on the fluctuations in electrical signals, these various bases were detected. So now this was one advantage over this third generation sequencing. And another advantage, which I would like to mention here, is regarding this reads, the range of the reads. So now we have already shifted into long read sequencing, wherein reads up to kilobase pairs of length can be sequenced in a single go, right? So these are various stages of NGS, and then how we have actually reached up to this point. Then next after this is, so this was all regarding your next generation sequencing, right? Then what about multi-omics? So multi-omics basically is a combination of various omics technologies, right? So where you can think of this as, say, epigenomics or genomics, then transcriptomics, proteomics, as well as you have various exposomes, right? So now I would like to give an analogy here, how we can think of these multi-omics approaches here and how they are related to each other. So we can think of this as building a house, wherein your epigenome basically serves as those zoning codes. So these zoning codes are basically those areas at which you can actually build your houses, the allotment of land, right? And then secondly, what we have is our genome. So this genome, you can think of it as a blueprint. So this blueprint basically serves as a master plan, that how the house should be built, like what should be the architecture, right? And then after this, what we have is our transcriptome, right? So you can compare this transcriptome as your building materials, right? So they actually involve in building that architecture of the house, providing those materials, right? And then finally, we have those proteomes. So proteomes, you can think of the ones who are actually bringing this house into shape, right? And you can think of them as your construction workers who are actually building those houses. And then last but not the least, we also have exposomes. So you can consider this exposome as various weather conditions, right? So obviously, due to various exposomes, our multi-omics or our genomic architecture also influences, and likewise, our susceptibility to various diseases also regulates, right? So this was a simple analogy that how you can think of multi-omics approaches. Then moving ahead, the next point which I would like to bring into the picture is the importance of these NGS and multi-omics data analysis. So the very first thing is how we have actually shifted into enhancing the resolution, wherein now at this particular stage, we have actually shifted our focus towards spatial transcriptomics, right? So for spatial transcriptomics, you can think of this as, you can see, maybe you have some various immune cells, and then you can see if you want to identify the macrophage, then the next question comes that where's the macrophage? So whether it is present in brain cells or lungs or heart or maybe in liver or kidney cells. So with the help of this spatial transcriptomics, we can actually we can not only identify the location, but we can also find out the expression, that what particular transcriptome or protein is actually getting translated, right? Then this is a simple example, a screenshot, a real images. On the left-hand side, you can see this is image for a simple light microscope, wherein you can just see that what all places your various proteins are getting expressed, or expression is being seen. But on the right-hand side, apart from getting the location, we can also see that what all proteins or what all transcripts are actually being translated at that particular time. So this is how this spatial transcriptomic actually helps us to do our biological data identification as well as further downstreaming analysis. Then the next thing after this is the power of this multi-omics integration, right? So like I mentioned, we have various levels of multi-omics, right? So then now combining those from a single perspective, we are actually getting more and more resolution and accuracy towards identifying various biomarkers, then maybe finding out the biological pathways, then also it is creating wonders in your drug discovery as well as therapeutic targets, right? And the most important point of which multiomics approaches are bringing into the picture is our shift towards this personalized genomic medicine, like right, wherein based on the genomic architecture, we are actually able to see that how we can provide personalized treatments based on the genomic profile of an individual, right? Then next thing after this is obviously this is the backbone of precision medicine, like the one I have already mentioned, right? So previously, what happens is we provide same treatment to an individuals which are maybe suffering from a similar disease, but there might be some biological biomarkers, different biomarkers, but similar kind of treatments are being provided. But obviously that may or may not produce effects. But with the help of this multiomics, we now first we examine something called as biomarkers, and then based on those biomarkers, the tailored treatments are being given, and which actually helps us to receive positive impact in all the individuals, right? Then next is our growing roles in research and industry, right? So the very first thing which I would like to point out is this Genome India project, which was launched by Department of Biotechnology, right? And recently, in the last month, the first outcomes were published. And in this, you can see from all over the different parts of the India, those human genome sampling were taken out, and then their genomes were sequenced. And then what is the interesting thing which has been identified? So here you can see so many, I mean, thousands of variants were identified who are actually linked with various types of disease, including your cardiovascular and metabolic disease. And then the most important thing out of this project, which came out doing your multi-omics data analysis, was identification of 44 million variants which are completely absent in all these reputed databases, including your Genome AD or 1000 Genomes project or Genome Asia. So you can see this Genome India project have actually profiled the disease scoring capacity. I mean the disease profile at the multi-omics level for all the Indian individuals. And now based on this data, the interventions can become more tailored, so we can get more tailored treatments, right? Then another thing is obviously the growing demand of this NGS and multi-omics data analysis role. So according to a survey from an market, it has been identified that by 2031, this market is going to reach around $27 billion, so which seems to be a very high amount, right? And then obviously the key drivers here are basically your advancement in sequencing platforms, and then rising clinical application, right? So there are so many opportunities here, including integration of AI/ML based technologies and adaptation of long read sequencing based approaches, right? Then the last point is why you should learn those skills. So the answer to this is in 3 points. The very first thing is high demand, right? So nowadays, not only in clinical research, but in agriculture and environment, everywhere, this NGS and multi-omics data analysts are required, having different set of profiles, right? And then the second thing is the salary is very handsome, and it is not only lucrative, but it also helps you to create more impactful work. And most of the time, the analysis which you do will be interdisciplinary in nature, and obviously you'll be solving the problems which are being faced by the world at present time, right? So with this, I would like to close my session. I hope I was able to bring some basic concept in you, and then we can, I would like to pass the stage to Urmimala ma'am. Thank you very much.
Thank you so much, Bhupender sir. That definitely gave us a very good idea what is happening around us, and it is indeed revolutionary. So we'll directly start off with the questions from our audience. So first up, we have the question from Subha Kannan. So basically, he is a 12th student, and he has this. He's from India, and he wants to join. He wants to pursue BSc Biotechnology, followed by MSc Bioinformatics. So very quickly, any one of the panel members, can you please guide him whether his choice is good or not, and whether it will be fruitful going forward?
Yeah, sure. Shall I?
Yes, sir, please.
All right. So, yes, definitely doing your bachelor's and masters in, bachelor's in biotechnology and then masters in bioinformatics is obviously a good choice. So, how it will help you out? So, obviously, when you will be going into the bachelor's, you'll start you'll be sensitized with what all dimensions are there, what all biology is there, maybe in the field of immunology, in the field of genetics, in the field of plant biology or environment biology. So you'll be exposed to what all dimensions are there, which can actually be taken into account. And then once you are having this idea, you have a little background of biology, then you can switch towards doing your masters in bioinformatics, wherein you will actually now learn that what all kind of data actually interests you, and then based on that data you can actually develop your bioinformatics skills, right? And for your information, I have also done similar kind of things. So although my academic background basically holds bachelor's in biotechnology as well as masters in biotechnology, but my project was actually the 6 month dissertation in bioinformatics was in, it was in bioinformatics. So the whole idea here is this will definitely help you in first building your basic biological skills, and then based on your interest in your biology, you can directly think of exploring that particular field utilizing your bioinformatics skills. Thank you.
Thank you, sir. Hope, Subha, so that you got a validation of your choice, and it's absolutely you're in the right direction. So continue to do that. Yashanji wants to know about the internships. Can you tell us internships which are there in this fields that 1 year student in 1 year the student can do? So Yashanji, we have a lot of courses with us, and you can explore our site. We do offer 3, 6, and 12-month duration projects. Okay, so our internship programs, it has a separate training period. So that is depending on the course, sometimes 30 working days, sometimes 45, sometimes 60. And if you're going for data science, then a 120-day working day course will be there, followed by the projects. Okay, so those project durations, if you want to go for a 1 year, you can select 12-month internship, wherein you will be getting guided mentorship with one of the expert panelists, and also we have other scientist members in our team. So they will be guiding you to do a dry lab project. So I'm getting some questions on wet lab that one of you have asked. Is there an internship from MSc Botany students to enhance the skills? So I'll come to that, but please be informed that we are talking here about dry lab internships, and these projects are going to be very easy. You can do at the comfort of your home, and you can easily complete them with just a laptop and some software and tools which your mentors will be guiding for. So this is Muskan's question. Is there any scope for MSc Botany to enhance the skills in molecular biology field? I request any one of the members to guide her briefly, as we have other questions also coming in line.
Yes, can I?
Yes, ma'am, please.
So, good evening, Muskan. I can understand your concern. You are from pure science background, and but you wanted to work with molecular biology. Even at Biotecnika, few courses we have MBTI, and then PCR related courses are there. Definitely, like see, basic molecular biology means like DNA isolation, followed by that PCR, and followed by that next latest advantage is like gel electrophoresis, and then then you need to shift off learning about some tissue culture techniques as well, and then some few microbiology techniques you need to learn, like how to handle in the laboratory, GLP practices. Along with that, then you can have a knowledge about like NGS or normal sequencing. So this will help you out in fulfilling your skills in molecular biology. I don't know, in your college you have some subjects and practicals related to that, Even in Biotecnika, few courses are there where you can pick up and do, or else like you can do somewhere at relevant research institutes for summer internship. You can go and you can gain these skills over there, Muskan. Yeah.
Thank you, ma'am. So hope that answered your question, Muskan. You can look for some wet lab experiences, and then definitely you can go for this dry lab internships, which is going to give you a complete experience. Okay. Okay, moving on to the next question from Anushka. I have a doubt. I am a second year biotech student. What kind of internships opportunities we can get for our skill development, and for MSc, which field is more advanced? So, if any one of the members can quickly give briefly an idea about what internships we offer and which one will be best for her since she is a second year biotech student. Nilofer ma'am, can you please guide her on this?
Yes, ma'am, sure. So I think as you have done the second year of biotech, right, and maybe you are pursuing accordingly. So the skills basically, if you're looking to like deep dive specifically with the bioinformatics or else biotechnology. So as the only thing which you need to be aware about the biological concepts, right? The practical skills which you can carry forward ahead, and if you're looking ahead with MSc with advanced fields in that specific career, so definitely we will highly recommend you to start upgrading your skills with understanding how the genomics data analysis can be done, right? How the multi-omics data sets can be used for the understanding the overall biological data interpretations. And with that, definitely you can start exploring the skills of basic bioinformatics, and then you can upgrade your skills in the programming also. So that is what is highly demanded right now. I think Bhupender sir have given right now a very good introductory presentation, and I think from there also you all of you get to know a basic pipeline how exactly you need to start, and skills exactly you can go with the multi-omics, genomics, and slowly you can start with the programming stuff. So I just all the best with that.
Thank you, Nilofer ma'am. We'll go to the next question from Dinesh. Ma'am, I'm a BTech biotechnology graduation just now, become a fresher. No one is giving opportunities. So, how to join reputed research industries? This is Dinesh question. Anyone would like to guide him, please?
Yes, ma'am. Shall I?
Yes, yes, please, sir.
Okay. So, as you mentioned that you have recently completed your BTech in biotechnology, and now you're actually looking towards jumping into the industry. So, obviously, the very first thing is the development of the portfolio, right? So, you have to create your profile in a manner that you can actually be more suitable to the particular position or industry roles which you're looking after, right? So, one recommendation from my side would be rather than going haphazardly, or maybe haphazardly in the sense maybe applying for 10 or 100 of industries without even seeing their job profiles, I would recommend you to first look into your skill sets and then think that what all positions are there and wherein you can go. And then now how you can jump into a reputed institute. So the very first thing will be to maybe you can join as a fresher. Maybe they, if they allow you to take as an intern, then that would be a great thing, right? Then this is going to be your one path. Then another path is like I mentioned, that you need to develop your portfolio, right? So how you can do that? So maybe you can think of taking an internship, doing some project works. So if I talk about your NGS and multi-omics data analysis, so you can do some course, you can actually do your projects wherein you will actually build your skills, wherein you can take up a research problem in your hand, and then you will utilize your computational skills to address that problem. And then similarly, you can actually post that into your personal GitHub repository or LinkedIn. And then what will happen? This will actually create credentials for you, which will actually certify that you have this confidence as well as skill set to actually address a research problem. So eventually that will set you apart from all other candidates who are actually looking towards getting into these reputed research industries. I hope I answered your questions. Thank you.
Thank you so much, Bhupender sir. So, I want to tell you that let's if you have specifically anything for NGS related careers, then our panel members are having great experience in that area. So, I would request the audience if you could ask more domain related questions. Okay. So I'll just pick up the next question from um, Ilma, I think. So, what skills to focus on if interested in pursuing research on aging, biology, or neuroscience? And yes, views on 4-year BSc, then PhD. Any advice on that?
So, good evening, Ilma. So if it's very good that you are thinking so proactively, so you wanted to work about do research on aging, neuroscience, and biology. Yes, of course, these are the factors really we are looking for in our modern style. Like we are, at the very young age, like aging is there, reflecting everywhere, and as well as a lot of neurodevelopment disorders are there, right? So if you wanted to pursue the research, so I would suggest like 4-year BSc is the basic foundation degree. Ultimately, the research means when you enter into the doctorate, you will experience the real crux of the thing. The for doing PhD or doctorate, the PSc will give you a very good foundation on thinking of, see, understanding the basic biology is more important, and understanding all the mechanisms is more important. So this 4-year BSc or 3-year BSc will give you some foundation. Then later on, if you can do like MSc, then you get into the PhD. This is the right order of doing. Definitely you will excel more in your PhD if you have the strong foundations skills you have. And one more suggestion I would like to give you, Ilma. Nowadays, like to do research, not only doing the wet lab matters a lot. Computational skills are also really matters a lot in doing advanced research in these skills. So I would suggest you to get a all-rounder when you are from either BSc and MSc, then do a doctorate is a right suggestion from my end, Ilma. Hope it clears your doubt. Yeah.
Thank you, ma'am, for the suggestion. So we do have a question here with choice of universities. We won't be, this is from Ishpreet. So we won't be able to comment on the university, but you have also asked on what are the courses that you are interested in. Should you go for BTech Bioinformatics, or you should go for BTech Biotechnology in specialization with computational biology? So that part we'll definitely answer. Anyone of the panelists would like to take up this question?
Yes, ma'am, I will take.
Yes,
So,
ma'am, sure.
so as you are willing to go for the bioinformatics career, right? So what we recommend you that BTech in bioinformatics will give you a straightforward domain expertise and try to upgrade more skills with respect to the bioinformatics stuff. So maybe in the BTech you will get specifically with bioinformatics a core specific domain expertise you can upgrade. Maybe like genomics designing pipelines, or as the multi-omics data analysis, how you can do, or else how to use the Docker or Nextflow. These are the very automations kind of tools available. So in this way, you can go very straightforward and then upgrading your skills from foundation level to the upgradation level. So that is how I think BTech Bioinformatics will provide you that roadmap. Whereas if you are switching to BTech in biotechnology with specialization of computational biology, so hand in hand you will get an experience working with biotechnology related fields. In that you can work with respect to the environmental expertise, right, or else with respect to some biomedical research kind of stuff. So you will have that kind of multiple roadmaps open for you. And in that, if you are willing to use that specific domain with integration of computation analysis, so that choice will be available for you. Whereas in BTech Bioinformatics, you have a one window access where you can upgrade the skills accordingly and then try to achieve your goals. So I hope that this two comparison you get the clarity. So if you're willing to like try to use BTech biotechnology with multiple domain area and then try to integrate with computational expertise, so I think that is also a good choice where you can proceed. All the best.
Thank you, Nilofer ma'am. So we have a question which is similar. Shall we do biotechnology or not? So I think you are quite early in your field. And we have a similar question from Nikita. Okay, so I would request Nikita to please post your question only once because the other questions are getting lost. I hope you will understand. Okay, so after answering this question, since you both asked similar questions, we'll delete the rest of your repeated questions. So request you to post only one so that we can get everyone's question resolved. Okay. So Nikita is asking if I go for BTech Biotech, what will be the entry level salary? Please tell me. I am 12th pass student and want to get into bio field. Can I go for biotech with PCMC background?
Shall I, ma'am?
Yes, sir, please.
So, like the entry level salary, so obviously we cannot definitely comment on this particular thing because why? We cannot tell you a specific number because it depends upon the type of organization or the type of research lab or industry you are actually applying for. But I mean, from my experience, obviously you'll have enough amount. I mean, you'll have a handsome amount even at the entry level also. But the most crucial thing here would be that you'll be developing your skills, right? Rather than looking for the say these remunerations or the salary at the very first thing, you should. I mean, obviously, you should focus more towards developing your skills, cashing upon those opportunities, so that you can get more expert. You will get more expertise over your experience, and from there onwards, once you will have more experience, then salary will just become a what you say byproduct of what your experiences are. So I would definitely recommend you to focus on your skill set, and this salary kind of thing and your positions will actually be a byproduct which you will not have to think of. Thank you.
Yes, sir. Very rightly said. If you want to go for biotech, then the passion should be there, right? So instead of asking that whether you should do biotech or not, you should ask yourself, right? Because if you have the passion in this field, then definitely you can excel. As you can see, a lot of examples in front of you only. So they are all successful in their field, and you will have to put little bit of dedication over here. And definitely success will follow you. You will not have to worry about those things. As Bhupender sir said, this will come automatically. So let's move on to the next question. We'll take up this question from Jyoti. Am I audible to all of you? Please confirm if I'm audible.
Yes, ma'am.
Yes, ma'am, you are.
Okay. Thank you so much. I'm sorry for the interruption. So yes, we have the next question, Jyoti, who has asked. I recently completed my MSc in zoology from university. For my project, I worked on microbiology based topic, even though my background is zoology. So I am interested in lab and research oriented career. My project title was Comparative Study of Microflora in Gastrointestinal Tract of Indigenous and Broiler Chickens. So any further career advice that Jyoti can follow?
Shall I guide on this, ma'am?
Yes, sir, please.
Okay. So, Jyoti, as I can see that you have completed your project in doing some microflora, identification of microflora in gastrointestinal tract in various chickens, right? So, I would like to bend this question into a NGS and multi-omics data point. All right. So now, what you have actually done is you have identified the data, right? Not identified, but you have generated the data. Now you have that data with you. But what comes next? Does the biology stops here? No. The bioinformatics now starts here. So now once you have your data here, now you can actually start analyzing those data. You can apply state-of-the-art computational approaches using various available models, or you can think of developing your own models to find out those hidden patterns from the data which you have generated, and then obviously you'll be answering more and more biology, right? And if you got good results, definitely it can be published. It can also enhance your skill set, your profile as well, and you'll be reaching to more, reputed position. I hope I am able to answer this. Thank you.
Yeah, thank you so much. So we'll be just limiting to one question for one of our audiences because we have limited time and there are a lot of questions which are coming in now. So, which NGS domains are seeing the strongest demand right now? Very good question, SK Asheri. Wonderful question. Thank you for asking this. So, cancer genomics, clinical diagnostics, microbiome analysis, single cell analysis, or multi-omics, which is seeing the strongest demand now? Any of the panel members would like to throw some light on this?
Good evening, Ashri. See, what you have asked is every field is important right now. Everything is in spotlight right now. So, if you see the cancer genomics is in the forefront towards the healthcare. So, with even for diagnosis, prognosis, the treatment, therapeutic response, treatment response, it's on the forward forefront. We are there. Like NGS is playing a huge role over there, and as I said. As a part of it, not only in cancer, like everywhere in the diagnosis stage, NGS plays a huge role. When it comes to microbiome analysis, this is also a recent trending field, I would say, because we have a microbiome, within us, good microbiomes we have, and harmful as well. So in the rare disease diagnostics, this plays a huge role, and as well as to understand the mechanism and how the novel targeted therapeutic approaches can be given for the diseases. That is also in the trend, I would say. Signal cell analysis along with the spatial cell transcriptomics, it's worldwide, it's having a huge scenario it's having now. Okay, not like bulk gene expression level, even at the single cell level, how the expressions are there, how the variants are there, being diagnosed not only for diseases, moreover in every part of the mechanism to understand. It is bringing a lot of revolution together with spatial transcriptomics. It is bringing on all the whole. You yourself has asked multi-omics. Yes, learning one layer itself, one layer of omics technology will give a more depth insights. When it's come to multi-omics, it has a wholesome idea. Ashri, every topics what you mentioned is more important. NGS and multi-omics have a huge role. If you are, if you have all those developed skills in this particular field, definitely you can shine over with good opportunities, and demand in the job is there. So if you have your skills in forefront, every employers will love to hire you, all of you. Yeah, thank you.
Nice answer on that, Ilamati ma'am. Thank you for explaining. Yes, so next question is from Rohit Kamath, recent grad in MSc Neuroscience with thesis on exploring metabolomics in Alzheimer's via NMR. What approaches would you recommend on self-learning concepts using real-world data to build a portfolio? Again, a very important question. Thank you for asking. Yes, any one of our members would like to answer this question, please?
Good evening, Rohit. Yeah. So you asked a very valid question, because nowadays industries look for the person who is expertised in the real world data analysis, because raw data really it is in a messy state. Okay, so who can turn into a biological, beautiful interpretation out of it, biological insight, developing a pipeline to analyze that, people's really look for. I know, like definitely on your thesis, when you have worked with metabolomics and also especially in Alzheimer's, you have put in some efforts in analysis. So when you are planning to do self-learning concept, no, you don't have to metabolomics. Okay, so in nowadays we have lot of biological databases with clinical data sets, are real world data sets are available. Definitely NCBI, Geo, and much more is there. So you can explore all those real world data, and you can try to learn from scratch with Python and R, and you can able to do analysis. And definitely with small projects you can develop in Kaggle and as well as GitHub portfolio, and you can make on your own. But learning the right thing with a proper stand takes sense. Okay, even though if you establish yourself with a your own projects, but you have some certificate in your hand, makes you valid in front of the employer that you are confident, along with the necessary skills you have taken and as well as you have experienced on yourself. But immediately to start with, I will suggest that lot of pipelines are already available in Kaggle and GitHub, so you can pick up, and as well as from the databases you can pick up your own data, and with your curiosity mind you can learn and doing on. But having said that, if you have certificate in your hand, that also makes sense. Yeah, hope you understand, Rohit. Yeah.
Thank you, Lamati ma'am, for throwing light on that. So I'll take the next question from Tawhid. I have completed RNA-Seq project, performed differential expression analysis and functional enrichment pathway using analysis using R and Linux. What else should I integrate to get an entry-level job? Dr. Nilofer ma'am, will you be able to
Yeah.
guide him on this part?
Yes, ma'am, sure. So great to know that you have done the particular project on RNA-seq analysis, and you have also moved ahead to perform the differential expression analysis. So I think you are very well know about like in the differential expression analysis, basically we used to get the genes which are differentially expressed with a specific disease which we are targeting, followed with the enrichment analysis, right? So and then you have upgraded your skills with the programming stuff with R and Linux. So what exactly we recommend is nothing but integrating like the skills which you have with handling this kind of big data, followed with the R and the Linux programming skills. So the entry level jobs is basically where you can get to know how to analyze the genomics data. So you can get certain kind of jobs where you can call yourself as a genome analytics. Okay, so you can analyze that kind of genome data. And moreover, if you have this straightforward skills with respect to the portfolio, which Elamati ma'am have already highlighted on that stuff, like creating your GitHub profiling profiles, or else posting your particular projects on the form of Kaggle or the GitHub, will help you to specifically upgrade your skills and get confident in that overall project and the skills what you have. So in that stuff, the entry level jobs, definitely you will get to analyze that kind of data, and if you're showcasing this kind of portfolio, then definitely you can even call yourself as a genomic scientist, where a comprehensive end-to-end screening you can do, and followed with that you can even interpret that kind of results with core kind of tools and the computational pipelines which you're trying to integrate. So that is what I think you can proceed.
Okay, thank you, ma'am. Hopefully that answered your question, Tawhid. So I think this question is directed to Bhupender sir from Anjali Gond. Sir, I have completed MSc Biotechnology. What NGS can help me in this? Please help me out for career opportunities in NGS, as I know coding knowledge is required in NGS field. Is that true? What should I do?
All right. So yeah. So since you have completed your master's in biotechnology, and now you are looking towards maybe shifting your career towards completely towards NGS data analysis. So maybe during the course of your masters, you had got some wet lab experience or idea of how NGS works in terms of library preparation or how you go for sequencing. Then now, if you want to shift towards the NGS data analysis or multi-omics data analysis side, so the very first thing for this particular to shift into this particular NGS and multi-omics data analysis stage is to understand the data. I think which you have already done that, and then now you can actually start exploring the computational part or bioinformatics part. You can start exploring various open source databases, including NCBI, or if you want to go into cancer genomics, then you also have TCGA, the Cancer Genome Atlas. Then what you will get there in those databases, you will actually come to understand that how the data is organized computationally, why structuring is required, and why there is a need for these sophisticated programming skills. Or nowadays, we also have something called as vibe coding, wherein we don't have to explicitly trained or be highly professional in coding, but yeah, some sort of knowledge is obviously recommended in order to evaluate what is being generated. So by first maybe start exploring the related databases, then trying out the already available pipelines or models or codes which are already published, and they are most of them are already available in your GitHub in their GitHub repository. So what this will help you? This will actually help you build a confidence in how the data is being analyzed previously, and you will also get a sense of how biological assertions are made based on the output which you are receiving from these tools and techniques. And then once you have got this confidence, then you can start taking up your research problem in your hand, and then maybe you can also identify your own gap in a various particular field, and from there onwards you can start building your skill set and getting more and more answers from biology using your NGS analytics skills. And regarding your question on coding knowledge, whether it is required in NGS field or not. So obviously, if you're going into data analysis, NGS data analysis part, coding knowledge is required, but that should not be as extensive as it was maybe in the past decade, because nowadays you also have various generative models or vibe coding, like I mentioned. So this will actually help you out doing most of the things, but that doesn't mean, like I mentioned, that don't have to have various knowledge, but you should have at least a basic knowledge where you can actually evaluate what is being generated, and maybe you can further guide on how you can make those programs more sophisticated. So finally, the guide would be to start exploring the databases, then start looking towards whatever all the models which are developed, then try to replicate them in your own platform, and then from there onwards, you can actually work on real world data sets. Thank you.
Thank you, sir, for the detailed answer. Hopefully that was a big help for Anjali. Next question is quite interesting from Lakshi Singh. I get to hear that I'm too young for NGS wet lab practices as a third year biotech student, but I want to learn NGS. I enrolled for a week-long NGS course, but everything it taught was already taught to us in the university. So what are the steps I should take next, as I want to be exposed to the labs and tools before I graduate? So we have a similar question also from one of the participants who wants to know how to understand the NGS platforms for skill development. So Pratibha has asked this. So if someone can throw some light on both the questions combined.
Shall I, ma'am?
Yes, sir, please.
All right. Coming to the first question, which is asked by Lakshi. So, that now you want to learn more about the NGS wet labs and the tools which are there. So, definitely, my personal recommendation for this would be to start looking towards various research lab in reputed institutes, like which are publicly funded institutes. So there, what they provide summer internship also for entry level students. So from there, you will actually get a hands-on exposure to how a particular various NGS tools are being used, or how you prepare a library. And you will also have a research environment there where you will get sensitized to how not only process the data, but then how analyze it using your various bioinformatic tools, and which will actually help you out in progressing further, that too with more real world experience. Then the next question, ma'am, can you please go to the next? Yeah.
Yes, sir.
So it's regarding how to understand the NGS platforms for skills development. So the very first thing, obviously, is to look towards the biological part, right? And in terms of NGS, you can think of first learning about how NGS system works, what are various library preparation techniques, or maybe how you go for, say, genome fragmentation, and then cluster generation, and then sequencing technique, right? So you will get so many resources which are already available there, which you can, that too from those companies who are actually producing these NGS instruments, right? One of them is Illumina, right? Then from there onwards, you can start looking at, like I mentioned, various research papers which are already have done some analysis through the help of NGS, right? So I have mentioned during my talk that Genome India program, right? So you can explore that page also. There you will come to know that how actually NGS is being employed to know this multi-omics profile specific to Indian population, right? So that will actually help you get more realistic idea. And if you want to gain more experience, definitely I would recommend you to start doing some project works, right? So that will also help you in gain more realistic real world experience. Thank you.
Thank you so much, Bhupender sir, for the elaborate answer. So we would be taking up last three questions for the day. And so this question is from Likhita. Is it okay if we go to some private university and learn some skills like AI, QAQC, or wait for some central universities for MSc Biotechnology? So, Likhita, you don't have to wait for anything. It can go parallelly also. Okay, so for all of your information, we Biotecnika do offer courses on everything AI, ML, all aspects. AI, ML in biology, AI, ML in chem informatics, AI, ML in drug discovery, whatever are the recent trends in the life science domain, research domain, we do offer. We also have QA/QC courses as well. So why don't you join in your MSc Biotechnology and parallelly run this, keep on taking these internships, keep on gaining experience. It keeps on adding to your resume and do projects. That's the most important because after all, your portfolio will matter a lot, right? And we do have a lot of questions. I mean, the same question posted a lot of times, although I requested, but then, so let's take this up from viewers. What should I do in my BSc Life Science to get ready for MSc Bioinformatics abroad? Even though it is not NGS specific, but we will take up this question as it is some light on abroad opportunities. So, yes, anyone would want to guide him, please, on this?
Good evening. Nowadays, like everybody wants to have the course in abroad or post graduation in abroad because lot of scope is there. Like due to that, every student have this interest. I would like to give some genuine tips for them. So when you are planning to apply for the colleges in abroad, first decide the country, which country you wanted to apply on. Moreover, there you pick up the universities who is providing a full scholarship and as well as who is providing tuition fees and who is providing full scholarship or partial. All these details you get into. If you are doing a BSc, you should start this search from first year onwards, okay? So then only you will land up, but after 3 years you will get trained up, then you will end up, and you can simultaneously start which country you are picking up. Based on that, IELTS is the most basic English exam test, as we are not non-native English speaker, so we should pursue that definitely as an Indian citizen. So start preparing from the BSc from first year onwards. Please prepare for these exams as well. And if you are going for trying for some other country, GRE, TOEFL also, you can prepare. But IELTS is very basic exam which is accepted by most of the universities. You can please do that. And moreover, people look for publication. If you have publication, that ad makes sense to you. And as well as what are the skills you gained, like practical skills and knowledge, clarity, what you have. That they will look for in your CV. And a statement of purpose, you should develop. So you might have a lot of interest, okay? So which course you are interested in, keep aiming from beginning only. So which college you need to apply, list you have it. So accordingly, when the application starts, keep an eye on that. And if you are applying beginning, then only you'll get the scholarship. I hope if you follow this, and publication is most matter. If you have one or two publication, and if you have good internship, if you participated in workshop and those things will also add some mark to you. And to get an offer letter from any of the countries from abroad, I would suggest please go somewhere with full scholarship. So that, because living in outside country, you need to invest a lot. So I hope if you do this, if you can prepare from the BSc first year onwards, at least by second year onwards, you will land up somewhere, like after completing your BSc, getting some good offer for MS or MSc in abroad. You can get, yeah. I hope this helps
Thank
you.
you. Thank you so much, Elamathi ma'am. So we have a question from our other platform, and this will be the last question that we'll take up for the day. Which programming language and computational skills should students prioritize first? Python, R, Linux, SQL, workflow systems, and cloud computing. Nilofer ma'am, would you be able to guide on this,
Yeah,
please?
yeah, I'm sure.
Thank you.
So great to know that you're willing to upgrade your skills in the programming stuff. Okay, so what I just highly recommend that as a beginner level, it's good to start with the Python programming, because in Python we have a step-by-step kind of environments ready with us. One of the environment which is called as a Jupyter notebook. Okay, so it's kind of the notebook environment. So it will give you a cell by cell writing of your command and understanding that command and getting the output. So then and there we are able to understand that what the command is written and what kind of output we are expecting. So in this way you will get the step by step understanding that how exactly the programming environment is all about. And followed with that you will come up with respect to the proper interpretations of that kind of commands or the codes what you are writing. So it's better to start with Python, and then slowly you can move ahead with R programming, because R programming is a such of the environment which is providing us the end-to-end pipelines. For example, if you guys are willing to proceed with the multi-omics screening or the genomics pipeline, proteomics pipeline, so we have end-to-end pipeline. End-to-end pipelines are nothing but when where you can start your data acquisition part and do all the in between the data analysis, pre-processing, the quality checks, right, normalizations of your data, and then making the data curations, because that is what important. So this entire pipelines is get created in the form of the R programming, and lot all the kind of packages are get involved in that. So in this way, after understanding what is the basic of Python programming, you guys will understand okay how this end-to-end pipelines look like. So in this way, from the basic to the integrated way, you will understand the overall cycle of that particular pipelines of the with respect to the programming skills. And while you are doing this kind of trial and errors, you will get a very great opportunity or as understanding about how to handle this kind of raw data, use the multiple packages programming stuff, and then understand your skills with the programming context. Whereas the SQL kind of stuffs are all about like going for the data which can be stored in the structured format, a big data sources wherever you have done your data analysis, and how you guys can proceed for the storing that data, and then using that structured data again with the programming stuff. So it's kind of the loop where you can start and then use kind of that kind of data and then switch with respect to the programming. So start with Python, upgrade your skills with R, and definitely this MySQL and kind of stuff will help you to integrate your data and that stuff. I hope that clears your question. Thank you.
Thank you, Nilofer ma'am. So, ma'am, if you can quickly guide on this question also, Nilofer ma'am, because the student has requested a lot of time. Currently, I'm pursuing BTech Biotechnology as a first-year student. My college is a three-tier college. My dream is to become a scientist. How should I plan my 4 years to be successful in my life? I think we have covered this
Great.
briefly from another question, but yes, personalized, if you can give him some guidance.
Yeah, I think from now itself you have that target to become a scientist. I think that is really good. Okay, so you have already planned your target. So the only thing is that you need to use the proper roadmap and how exactly it can be get achieved. So what we suggest you accordingly from our end is that having your like the skills with BTech in biotechnology. As I mentioned earlier, that BTech biotechnology will provide you a multiple domain areas, okay, like the biomedical, okay, then the environmental kind of stuff, biotechnology itself, and followed with that bioinformatics is even integrated with BTech kind of courses. So try to understand like which particular domain you want to like deep dive in that, okay, and you want to perceive in that particular career itself, so that because in that stuff you can call yourself as a scientist. Okay, so the plan what you can do for the fourth year is that you can definitely proceed ahead to pick up some project. You can welcome to Biotecnika, okay, you can take some online or as offline project. You can plan for the fourth year, maybe taking a 3 year or the 6 month of project. Okay, in that stuff you will get a very personalized kind of training. Followed with that you will get to know how to use this overall skills and the knowledge to design an accurate project pipelines. And in that stuff by the end definitely you will get some kind of conclusory kind of findings based on the objectives which you are providing, and followed with the guidance whatever the Biotechnica team will provide you with the technical level as well as with the skills level. I think that experience will definitely help you to get smoothly landed to call yourself as a scientist. So great, good to know, and all the best once again.
Thank you, Nilofer ma'am. So I am very much tempted for this one last question because I'm sure some of you here will resonate with this as well. So I'm going to take up this last question, very last, final last, from BT Neha. I would like to know whether a career in NGS would be suitable for someone returning to the biotechnology field after a long career break. How to get remote role after NGS theoretical course. If anyone can please guide, because many of our students, they are on a break for some reason or the other and they want to come back. So how can NGS help them get back on the field?
Shall I, ma'am?
Yes, sir, please.
All right. So, yes, having a long career break obviously not only compromises with your skill set or whatever things you have learned, but I mean, this also has a positive side. You can also start afresh, maybe choosing some other domain, coming with different mindset, right? So now coming to the point that how you can actually shift towards NGS. So the very first thing, like I mentioned in my talk as well, that this NGS and multi-omics field is continuously evolving and it is exploding the entire biology field. Say it's whether it is human genome or biotechnology or agriculture or environment, everywhere this is present. So obviously now if you have huge amount of say demands, so obviously there is going to be so many organizations who are also offering good good roles, as well as training the students to get those roles, make themselves more eligible to those roles, right? So now coming to your next point, that how you want to how you can get those roles. So obviously the very best thing would be to develop your skill set, start taking up projects, develop your portfolio, and then apply in more targeted roles. In the sense, try to apply in those positions which actually matches with your skill set. So that will actually load down your what you say, the frequency of your applying to position, and it will also help you get more time when you are applying for a particular position. And then if you're looking towards remote roles, so what I remember is we have EBI, which is your European Bioinformatics Institute, then Uniprot as well. They are actually offering these remote positions of, say, NGS data annotators, wherein you actually have to annotate the data from the research papers or review papers, or you have to create a database. So, or maybe you have to validate the model's responses. So all those things are being offered in a remote manner. So you can think of taking up those positions, as well as some industries, as well as various. Nowadays, you can see AI companies are actually jumping into this biology field as well. So specifically, they are also looking for remote positions where you can actually contribute towards data annotation or labeling or benchmarking their model. So many opportunities are there. So, but my last thing for this particular question is start building your skills, right? And then once you have your skills built up, then start taking up some projects. And then after taking up those projects, definitely you'll be in a position wherein you can post yourself as a expert who is having the capacity to analyze that data, and that will help you stand apart from all other applicants, and you will definitely be able to secure a reliable position. Thank you.
Thank you, sir. Thank you so much. So with that, we come to an end of our Q&A session. And thank you so much to all our panelists for patiently answering all those questions and for sharing your valuable insights. I'm sure many of you now understand why NGS and multi-omics data analysis is becoming such an important career domain, not only in India, but globally. So it is no longer limited to wet lab experiments alone. Modern research and industry, they are increasingly driven by biological big data, sequencing technologies, AI driven analysis, and computational interpretation. And because of that, students who combine life science with data analysis skills are getting a major advantage in research career, biotech company, healthcare analytics, precision medicine, pharma, and also AI driven biology startup. So keeping this growing demand in mind, Biotechni- Biotecnika is launching its upcoming course on NGS and multi-omics data analysis training plus project program, which is starting 14th of May 2026. This includes hands-on practical training with guided projects, as I mentioned before, training separate, followed by projects, exposure to real analysis workflows. You'll get mentor support, personalized guides to guide you, mentor you through your project from working scientists and industry oriented skill development. So, if you are serious about building a career in bioinformatics, NGS, computational biology, or AI in life science, this is definitely a domain worth exploring deeply. And now it's time for the surprise gift we promised at the beginning of the session. So, for all those who have stayed with us till the end, we are sharing a special resource link in the comments section right now. Please copy and save it quickly. We have also shared our links if you are interested in our in the course of NGS. So you can click on our store page. The link is given over here in the comment section, and the link of the free gift is also given at in your comment section. Do copy it, otherwise you will not be able to access it once this live session ends. So this resource includes NGS career guidance, beginner roadmap, important tools to start with, and learning direction for students entering this field. So make good use of it, because honestly you usually don't get such structured career guidance handouts easily online. So don't miss it. Okay. So before we conclude, I would like to sincerely thank to all our respected speakers, Dr. Bhupender Singh, Dr. Elamathi Natarajan, and Dr. Nilofer K, for taking out time and sharing such practical and valuable guidance with all of us today. And a huge thank you to all the participants as well, because you asked so many important questions and you made the session truly interactive and engaging. I'm sure today's discussion has given many of you a much more better clarity. So if you genuinely found this session useful, do share it with your friends, classmates, give us a thumbs up, and colleagues as well, because many students are still unaware of the opportunities available in this field. So before leaving, please do share your feedback in the comment section. We would really like to know what you found most helpful, what kind of future mentoring sessions you would like us to organize next, and if there is any other suggestion. So thank you once again, everyone. Have a wonderful day ahead. See you all soon in another exciting scientific session. Good night.